Hi, everyone. Welcome to the Johns Hopkins Cerebellum Seminars. Today, we're hosting Catarina Osório. Catarina received her PhD in neuroscience from Cardiff University, working on the molecular mechanisms that regulate axonal and dendritic outgrowth. She was mentored by Alan Davies. Then she went to King's College in London, where she was mentored by Oscar Marin and Beatriz Rico, where she further developed her studies on the molecular mechanisms required for interneuron migration and synapse formation during development. As a postdoctoral researcher now in Martin Schoenville's lab at Erasmus MC in Rotterdam, she's using a wide range of techniques to investigate the role of cerebellar genes expressed during development. Now, these genes are necessary for correct sensory motor integration. Today, we're going to learn about her work on the intrinsic activity of Purkinje cells and their role in the development of the cerebellum.
Thank you, Reza, for the kind introduction and also for giving me this opportunity to share my research with all of you today. So I'm a molecular biologist, and my research focuses in understanding the mechanisms underlying neuronal development and ultimately neuronal function. And why do I think development is or neurodevelopment is so interesting to study? Well, you can see here during the human development timeline, this is a long period, and there are several neurological processes that need to occur at precise time points. We have neurons being generated, migrating to their final destination, axons and dendrites are growing, and connections are formed. And I hope you can see my pointer here. The third trimester of gestation is a particularly interesting time because many cellular processes occur at the same time. And of course, this makes this time point very vulnerable to genetic insults and environmental insults as well. It takes only one of these cellular processes to be dysregulated to lead to long-lasting deficits, underlying neurological or neurodevelopment disorders. But what is also interesting is that brain development or neurodevelopment doesn't occur equally across all brain regions. In this table, you can see the process of neurogenesis in the spinal cord and different brain regions. And of course, the cerebellum stands out by its prolonged developmental period, which again make this region susceptible to errors throughout all this time. But neurodevelopment is a process that requires the timely and precise coordination of activity and genetic genetic dependent mechanisms. And this is particularly relevant during cerebellar development, where billions of neurons need to be generated, migrate, and form connections. And as you can see here in this drawing by Ramon and Cajal, at the center of the circuitry are these beautiful Purkinje cells with large dendritic arbors that receive inputs from many, many neurons. But something that is also very interesting regarding Purkinje cells is that in the absence of synaptic inputs, they are able to fire spontaneously or be intrinsically active. And we have shown in our lab that in the mouse model, this intrinsic activity is already present at postnatal day 3 and then increases gradually during the first 2 weeks of development until it reaches adult stable levels. So we think that this period is very interesting because it suggests that intrinsic activity needs to be properly regulated, and perhaps it's even responsible for regulating different aspects of cerebellar development. Something also very interesting about intrinsic activity is that this function of this sort of activity in the Purkinje cell is present in several models, several mouse models of disease, such as cerebral ataxia or autism and neonatal hypoxia. And sometimes this dysfunction already occurs during the asymptomatic period. So Elena Watt's lab has suggested that this dysfunction or early dysfunction of Purkinje cell intrinsic activity is perhaps the drive that leads to disease progression. And our own work on mouse model of spinal cerebral ataxia as CAM1 has shown that there is a temporal sequence of events before the onset of ataxia. And these events start with disruption of intrinsic activity, but also followed by deficits in morphology. And just before the symptoms of ataxia, we observed that there were deficits in cerebellum motor learning in these mice. And then around 5 weeks, this model becomes ataxic, or at least we can see and we can test this. So with this in mind, we decided to dissect the role of intrinsic activity during development, and we wanted to explore that if indeed disruption of this type of activity in Purkinje cells would be sufficient to generate cerebellar pathology, and also what would be the role, how it would influence cerebellar development. So to answer these and many other questions, we have generated a mouse model that allows for temporal control of activity. And at the core of this mouse model is the inward rectifying potassium channel Q2.1. So if this channel is overexpressed in the membrane of the neuron, there is a hyperpolarization of the membrane potential and the probability of the neuron to fire is reduced. So our mouse model, we have an Purkinje cell specific inducible conditional Q 2.1 with an M-chair reporter. And if we inject low doses of tamoxifen, we can have sparse labeling, as you can see here. And this sparse labeling is quite useful if we want to analyze the morphology of these neurons. And as a control, we use inducible Purkinje cell specific conditional D2 tomato or AI14. For most of our experiments, we want all Purkinje cells expressing KIR 2.1, and for this, we either inject high levels of tamoxifen or we use the conditional PCP2 KIR. And with these mouse models, we first have to validate them if they indeed were silencing intrinsic activity in Purkinje cells. And to validate our models, we did both in vitro and in vivo experiments. So as you can see, one of the first experiments we did was injecting tamoxifen in our inducible mouse models, both control and mutant, at postnatal day 1. And then we wait until the whole developmental period passed, and so 3 weeks of development, and at postnatal day 21, we did slice recordings. These slices were in the presence of synaptic blockers, and most of our control cells fire spontaneously, were intrinsically active, while most of the mutant Purkinje cells were silent. But despite the loss of intrinsic activity, these neurons were still excitable. So if we inject current, these neurons are still able to generate action potentials, but we needed to inject around 600 picoamps or more. But nevertheless, they were excitable. So the in vivo experiments we did around approximately postnatal day between 28 with our PCP2 KIR conditional model. And as you can see, the control Purkinje cells have the typical simple spikes and complex spikes, but the mutant Purkinje cells show a reduction of the simple spike rate and an increase in irregularity. But it was also quite surprisingly, at least for me, that the complex spike rate was increased compared to the control. So overall, Collectively, this data shows that by overexpressing Q2.1 in Purkinje cells, the intrinsic activity is reduced, which was what we were aiming for. So with this, we start answering some questions or testing some of our hypotheses. So first, we study how disrupting intrinsic activity from early development could influence Purkinje cell morphology. For this, we use our inducible models. Again, low doses of tamoxifen were injected at P1, and after 3 weeks, we analyze the morphology of these neurons. So I hope you can appreciate the difference between the control neuron and the mutant Purkinje cell, how different the dendritic arbors are. We quantify this with shell analysis and can appreciate the complexity of the dendritic arbor in the mutant cells is reduced, as well as other parameters such as area or the longest dendrite. Then we use our conditional mouse model to study the number of inhibitory contacts Purkinje cells were making with their cerebellar nuclear cell target cells. And for this, we count the number of inhibitory photons that were colbinding positive and VGAT positive in the around the perimeter of the cerebellar nuclear cell. And you can see the number of presynaptic inputs in these neurons were reduced in the synaptic inputs made by mutant Purkinje cells were reduced compared to the controls. And we record these cerebellar nuclear cells in vivo, and you can see that the firing rate of the mutant in the mutant mouse is significantly increased, as well as their irregularity. So overall, disrupting intrinsic activity in Purkinje cells during the entire developmental period impairs the dendritic arborization, the formation of the dendritic arborization, and the inhibitory contacts it makes with the cerebellar nuclear cells. Okay, so we then asked how disrupting this developmental period until adulthood could influence behavior, and we did a battery of tests. We allow our conditional PCP2 KIR 2.1 model to grow until adulthood, and then we start by doing the balance beam test. So this test was done in a 12 millimeter flat beam, and you can see that the mutant animals are taking longer and made more missteps compared to the controls. It's actually quite challenging for them, and when we quantify this, you can see what we see in the video. And when we challenge the animals with a 12 millimeter round beam, 10 out of 10 controls were able to cross the beam, but the mutant animals were unable to cross. We analyze interlimb coordination, and we what you can see, and we also get here this plotted, is that the distance of each step made was not different between control and mutant, but the time of each step was actually longer in the mutant. You can see that sometimes the mutant animal has the paw in the air before putting back on the floor. So this step is taking longer, and also the body axis swing is shifted in these animals compared to the control. So these are some features or deficits in balance and coordination that also shared by some ataxic mouse models. But here our model is disrupting intrinsic activity well, from very early on during development. And these are our last behavioral tests that were cerebellar dependent motor learning tests. So we do our favorite in the lab that is based on the vestibular ocular reflex adaptation. So these VOR or these vestibular ocular reflex allows to stabilize the visual field when our head moves. So the eyes move to the opposite direction of the head. And in the lab, you can see we can do that with a turntable that is mimicking the vestibular stimuli, and the drum is mimicking the visual stimuli. So we use a phase reversal VOR adaptation paradigm as a learning test for our animals. So what the animals need to do here is reverse their VOR. So they have to move their eyes in the same direction as the table. So visual and vestibular input are in phase. And you can see the control animals, they learn this task very well over the course of 5 days, but our mutant animals were unable to learn at all. Another favorite in the lab is the eye blink conditioning. So in our case, we use air puff to the eye as an unconditional stimuli, and the animal has a reflective eyelid closure, which is the unconditional response. And we use the LED lights as a conditional stimulus, and after pairing the stimuli for many times, the animals start learning to have a preventive eyelid closure when they see the light. That it's what we are quantifying the conditional response. And similarly, the control animals learned this very well over the course of 5 days, 2 sessions per day, but our mutants were unable to learn. Okay, so overall, our behavioral data tells us that this disrupting intrinsic activity from development prevents the acquisition of balance, coordination, and cerebellum motor learning. Well, we next were curious to understand if there was a temporal window of action for Purkinje cell intrinsic activity during development. The data I've shown you so far, what we did was disrupt activity during the entire developmental period, sometimes until adulthood. So in our next experiment, we disrupt activity in Purkinje cells only in the first week of development. So injecting again low doses of tamoxifen in our inducible mouse model, and at P7 we study the morphology of the Purkinje cells. So here you have example images, and the mutant Purkinje cells had a very different morphology compared to the controls. So this morphology is basically several neurites coming out of the soma compared to what you can already see. It's a baby Purkinje cell. It looks like a Purkinje cell, it's just smaller. It has a 1 or 2 apical dendrites with a flattened dendrite. What was interesting in this result is actually this phenotype belongs to the normal development of a Purkinje cell. It was what Monica Hall has described as a stellate cell with disoriented dendrites. In fact, he made a drawing about all these time points of development of Purkinje cell, and you can see in A what these phenotype, multiple neurites coming out of the soma, and here you have the example of the rat Purkinje cell development. And after A, the neurites retract, and there's usually 1 or 2 that become this apical dendrite. So what we are doing, it's by disrupting activity during this period, is delaying maturation. So next, we disrupt activity in the second postnatal week of development, and you can see that the mutant Purkinje cells are also smaller and with a less complex dendritic arbor compared to the controls. And we finally did this experiment, just disrupting intrinsic activity only in the third postnatal week. So there were differences in morphology between these control and mutant Purkinje cells, but these differences were not as severe as the previous ages. And if we actually compare this with the disruption of activity during the entire developmental period, you can appreciate the difference. So we think our data suggests that the requirement of intrinsic activity for Purkinje cell morphological development is decreasing with age. Okay, so we wonder if these requirements or time-dependent requirement could have any relevance in behavior. So we went back to the balance beam test. And here you have the result that I've shown you before when we interrupt the entire developmental period until adulthood. So now we decide to use our inducible model to inject tamoxifen at P7 or at P14 or at P21, and now let the animals grow until adulthood and then test them in the balance beam. So we are skipping disrupting activity 1 or 2 or 3 weeks. And overall, these are the results. So what we can see in these graphs is it doesn't matter when we disrupt intrinsic activity of Purkinje cells, the mutant animals will always perform worse than the controls in the balance beam test. However, the severity of the phenotype is reduced if the disruption of activity is delayed. So even though intrinsic activity is crucial for motor control, when this activity is disrupted during development makes a difference in terms of the severity of the phenotype. And the final question we were interested in answering was, what would be the activity-dependent molecular mechanisms underlying Purkinje cell early activity? And because we observe already changes by disrupting activity in the first postnatal week, we decided to collect Purkinje cells from a conditional mouse model from controls and mutants at postnatal day 7, so after a week. And we run a bulk RNA sequencing analysis, and we did differentially expressed analysis, and we had around 1,600 differential expressed genes. And these genes were in 3 main groups in terms of biological processes. They were involved, these differential expressed genes were involved in the regulation of the nervous system development. There's 2 nodes that group these genes, and there's a second group, a second big group that pertain to synaptic signaling or synaptic transmission or plasticity, and another group was the calcium ion transport or calcium signaling. So these were the main biological processes that our differential expressed genes would follow. And after this, we also decided to run KEGG enrichment network analysis to understand which signaling networks were being altered in our mutant Purkinje cells. And again, here you can see there's some pathways that are changed, calcium pathway, phosphoinositol pathway system, and of course, long-term depression that it has a bit of both. And we also had many synapses and axon guidance genes, but also very interesting for us was to see that there was the pathways for neurodegeneration and cerebellar ataxia, Huntington disease. So there were very many interesting networks being altered. And because the phenotypes we had in our behavior were mainly motor impairments, we decide to extract a list from the CAG disease database that had to do with genes involved in genes that or mutations in genes that would lead to motor disorders with a cerebellar component. And we had around 216 genes in this list. And when we compare to our differential express gene database, we extracted 25 genes. Most of them were downregulated. And I think you can recognize a lot of them being involved in spinal cerebellar ataxic phenotypes, but also a lot of these genes are involved in mitochondrial function and a bunch of these signaling pathways. So we wonder if these genes could also be involved in early developmental processes, in particular regarding Purkinje cell maturation. And we chose the top 2 genes to test this. So the Prkcg gamma translates into protein kinase C gamma, and mutations in this gene lead to spinal cerebellar ataxia 14. And CAR8 translates into carbonic anhydrase 8, and mutations in this gene are known to lead to a disease that involves ataxia, intellectual disability, and also disequilibrium. So to test how if we change gene expression, we could affect Purkinje cell maturation. We used Purkinje cell specific conditional knockdown model. So to develop this model, we design shorter pins RNAs against each of the genes and against LAC-Z as a control. We package the shRNA in adeno-associated virus that also expresses an mChara reporter, inject them in neonates of a Purkinje cell-specific CRE, and after a week of development, we look at Purkinje cells. Here you have in situ hybridization showing the levels of gene expression in the control for the Prkcg gamma and the knockdown, and also in the mChara is expressed here, indicating these cells were infected, and here you have the knockdown of CAR8. Okay, so then we analyzed the morphology of these neurons, and we had some distinct effects for each of the gene. So the knockdown of Prkcg gamma led to an increased dendritic arborization, increased area of these neurons, and also a reduction on the number of neurites coming out of the soma. And this compared to the control, suggesting or indicating this gene is or this protein Prkcg gamma is a negative regulator of dendritic development, while knocking down CAR8 led to a more KIR2.1 overexpression phenotype with multiple neurites coming out of the soma, and you can see here, and we think this gene is important on the transition from this stage of multiple neurites to 1 or 2 apical dendrites that will make the typical Purkinje cell morphology. So these are just 2 examples, but there are many, many other genes that we haven't tested. But overall, this is giving us an indication that already in the first developmental week, intrinsic activity is leading to it's responsible for several transcriptomic programs that are regulating different aspects of dendritic development in this case. And so to summarize, we have shown that disruption of intrinsic activity is sufficient to generate cerebellar disease, but also intrinsic activity is important to regulate neuronal maturation and function in a temporal sequence of events. And this is modulated by a activity-dependent transcriptional program that regulates dendritic maturation, connectivity, and motor function. So, and of course, this happens at specific time points during development. And with this, I would like to thank the Schoneville Lab, in particular Martijn and Josh, and all the students that participate in this project over the years, and also the Department of Neuroscience, our collaborators and our external collaborators, and also the funding agencies that have funded this and many other projects in the lab. And I thank you all for listening.
Thank you, Catarina. That was just
Thank you.
wonderful. Let's ask if you have any questions, please just go ahead and raise your hand. I will call on you. Let me start, Catarina, by asking you to tell us a little bit more about these Purkinje cells in terms of their how they might be functioning during behavior. You showed that manipulation is clearly reducing the intrinsic activity, perhaps suppressing it completely, but you also showed that their response to input was significantly reduced. Do you think they're just like that they're going to be completely silent during behavior? What do you think?
No, I don't think they are silent, because I think they're still receiving some input from the excitatory inputs, but I don't know what climbing fibers only can do. And this is funny, because they also degenerate eventually, and yeah, it's really weird, because we are doing the extreme situation of reducing activity, but some activity is happening, and I think there's a few simple spikes, but there is complex spikes, so there is some activity going on. But yeah, I cannot explain what simple spikes, complex spikes only can do, because in other models of ataxia is not that dramatic. So this is a dramatic model.
Yeah.
So yeah.
Rich, you have your hand up.
Thanks for the nice tutorial. And I just wondered if you could speculate on, for those of us who work with patients, there's this striking sort of difference between, right, the early onset pathology versus late onset pathology. And much of the discussion centered on that maybe the early pathology somehow disrupts the organization of the cerebral cortex rather than resulting from disorganization or dysfunction of the cerebellum. And I didn't know whether you had any thoughts, or any of you
Well,
was looking at the rest of the brain to see what sorts of consequences or changes came about from these early insults or early manipulations.
well, I don't know so much about the clinical aspects. I know there are some eye movement studies that were done in carriers of ataxic mutations, especially I believe it was the SCA3. So it was the ataxin 3 mutation of known carriers, but they haven't developed the phenotype yet. And these eye movement paradigms or tests show that they could already see a difference in the carriers compared to the control. And as we saw in our mouse model, but this was again a SCAO1 mouse model that is specific for Purkinje cells. We think that during the the asymptomatic period, there is indeed some changes in the circuitry that are happening and build up over time. Now, I don't know if it's possible that the activity is not changed based on this. And so in terms of patients, of course, it's easier if you know they are carriers, but for people that work closely with this, and I know that some people have been trying to change activity to ameliorate the symptoms. So, yeah.
Right. No, I'm just to follow up very quickly. I was actually more interested about in the mouse models,
Oh, yeah.
whether you would see changes, say, if you were to look up in the cortex, what changes you might occur up there
You mean the
in these.
neocortex?
Yeah, yeah.
Oh, yes, yes. Okay. Well, that's very interesting because we know that if it changes in the cerebellum, we actually tried to do that very early on. I just think we didn't use the right tools to assess this properly at the time. But yeah, there are someone here is doing some of those experiments by damaging the cerebellum and looking in the mouse model and looking at the prefrontal cortex, I believe. And indeed, there are some changes, but at the level of the synapse. And that probably was my mistake with this mouse model. I didn't go to the level of the synapse to look at the spines to see if there were morphological changes in the frontal cortex, indeed. But some people are seeing that there are changes already. Yeah.
Thank you.
In the neurons. Mm-hmm.
Thank you. Stephanie, please go ahead.
Hi, Catarina. Very nice. I have two small questions. The first one is on whether you could speculate what is driving the early activity in Purkinje cells when the synapses are still very sparse or not even there. So do you have any hypothesis what could be driving this other than just intrinsic excitability? And the second question is about your gene network disruptions or changes in the knockouts. Did you perform this in a specific region of the cerebellum, or did you take the entire cerebellum?
So I start with the last one because it's simpler. We took Purkinje cells from the vermis and from different lobules. Yeah. Then the first question. Well, when Alana Watt was a postdoc, she has this nice paper with Michael Hauser that shows that from early development around postnatal day 9 in the mouse model, there is these collaterals of Purkinje cells that connect with each other, and they have these spontaneous waves they transmit to each other. And these collaterals are reduced in the adult. So that happens in other brain regions during development. There is this initial contacts that we don't see them in the adults, but they are transient contacts that are made very early on during development, that start the first activity in these neurons, and of course gene expression and development before there is connections with other neurons that haven't arrived yet to the circuitry. So there's that I think is a very nice paper, and it is the only one so far that I'm aware that shows something like that during development.
Do you think the synaptic transmission from the collaterals is still excitatory?
I think so. Well, until postnatal day 9, yes. I mean, there's this paper very early on in time for me that it shows that GABA is polarizing in Purkinje cells until around postnatal day 8, 9, and then it shifts. So I think so, yeah.
Okay, thank you.
Catarina, the behavioral results were so strong. I thought there was like zero learning
Zero.
in the couple of examples that you showed. For the VOR adaptation in particular, was it that in both directions, gain up and gain down, you see no learning at all?
Yeah, I don't have it here. Also, but however, our gain increase is not as it is not over days. We do it in 1 day, so it's also a different test to compare indeed. But we also didn't see any learning. And I was more impressed that in the baseline, I believe I have it here. Yeah, I was more curious that in the baseline, the difference were not, well, there was differences in the OKR, but I was expecting to see also a bigger difference in the VOR. There is a bit, but yeah, it was more when we challenged them with the learning.
Okay.
That was a problem, yeah.
Molika, you have your hand up.
Hi, thank you so much for the talk. Very amazing. I have a question. So, correct me if I'm wrong, but from what I understood, you showed that the later you disrupt the activity, the more severe you got the defect. This is right?
Oh, no, the other way around.
The other way around.
The earliest the disruption, the most
Okay.
severe the phenotype, yeah.
Okay, so because I was thinking about some sort of compensation that can be present in because in this disease we don't see this amazing big enormous defect after development or even during development in ataxia or in Huntington disease. So I was thinking, do you have any thoughts on what could be there as a compensation or more vulnerability even? I'm not sure which one it could be, but
I don't know if it's a compensation. It's perhaps how early or late these changes start to occur. And that I think that in this case, we are starting these changes very early, but in most human disease, first the time is different. And there's a long period that it leads to accumulation of errors that until you can see some physical changes. And that's when people start thinking about this. But perhaps there's a long period before where these changes are being made slowly, and we are hypothesizing it starts with activity. And this affects basically all the genes that are important for the morphology, that the same genes that are similar genes are responsible for activity of the cell. So any change is building up. And this is why I don't know if it's a compensatory mechanism more than here, this model is really extreme.
okay, thank you.
It's wonderful. I learned so much. Since we have a little bit of time, maybe I can ask you to speculate. One of the things you showed earlier was that the development of the cerebellum takes a really long time. For example, compared to the basal ganglia and the thalamus, peak neurogenesis in those regions is mostly finished in the prenatal period, while in the cerebellum it continues well after. In humans, it goes into late childhood, maybe adolescence.
Adolescence, yeah.
Yeah, so what do you think? And that sort of seems similar to the cerebral cortex to me, the development arc of the cerebellum and the cerebral cortex, and they take a really long time to form, whereas the basal ganglia and the thalamus, they don't. And so what do you think that says about the cerebellum?
Well, it has a lot of functions that is still yet to dissect. I mean, for sure, there's there is a lot. This is what I thought it was so frustrating when I came to Martijn's lab, because I kept asking him, oh, there's no papers about this. I'm not about the development of whatever cells. And then he was just, oh, well, because there isn't. There's no many, because I came from the neocortex field where like there's groups and subgroups of glutamatergic neurons and interneurons, and there's like a lot of research of different aspects of development. And indeed, in the cerebellum, we are very, very far to that stage. So, and of course, now we know it's more than motor, but even the little details about developmental programs, we are still unaware of them. So of course, the cerebellum might be involved in many other functions, and now we are learning more how it's connected to different brain regions. And so I think it's a very exciting field to be studying development, or brain regions to study development.
Yeah, I'm glad you're doing it, and I very much enjoyed learning about it today, Catarina. Are there any other questions for Catarina? All right, thank you, everyone.
Thank you for having me.